Good morning, everybody, and good afternoon, who is in the North Hemisphere. It is my pleasure to introduce you the student Lucas Rabelo de Vasconcellos, who is a candidate in the defense of his undergraduate project that was supervised by Dr. Musa Mhlanga from the Radboud University in the Netherlands and co-supervised by Dr. Élida Leite Rabelo from the Federal University of Minas Gerais. Today we will have as an evaluator Dr. Laszlo Groh from the Radboud University, also from the Netherlands. And then I just want to extend all of you our acknowledgements, and we are so thank you all for contributing for the formation of our undergraduate students here at the biological sciences course. Okay, my name is Lucia Valentina Modulo, the coordinator of this session. Then now I will just share Lucas' screen. Um, Lucas, you have up to 30 minutes to um, give an oral presentation, and whenever you're ready, you can start, okay?
All right. Okay. Good morning, everyone who is in Brazil. Good afternoon, everyone in Europe. My name is Lucas, as you're probably aware, and it is my pleasure to be here today telling you a bit about my graduate thesis project about the use of DCAS9 crab for the perturbation of the interleukin 8 topologically associating domain for investigation of spatial transcriptional regulation. And I was supervised by Professor Musa Mhlanga and co-advised by Professor Élida. To talk about that, we're going to need a bit of introduction first. First, I'm going to tell you a bit about gene expression regulation, and genes are controlled by intricate networks of regulatory elements. And they fine-tune gene levels according to context, which might be cell type, physiological state, a moment in time, for example. And in this scenario, enhancers are critical elements. They are DNA elements distal to genes. They interact with gene promoters through DNA bending and looping, as we can see here. They recruit transcription factors and co-activators, which in turn make it easier to recruit RNA polymerase to facilitate more efficient and more frequent transcription. And a big part of enhancers as well is that they are important for cell identity and differentiation because the scenario of active enhancers varies from cell to cell because transcription factors can be cell specific, cell type specific. So that's how we can have many different cells in the human body, even though they share virtually identical DNA. And so enhancers are pieces of DNA sequences, and DNA is in the nucleus of cells, but it's not just loose in the nucleus. It's actually organized in very specific structures. First, in a smaller order, DNA coils around the histone octamer, as we see here, forming nucleosomes. And then the nucleosomes are closely packed together into chromatin fibers. And then this chromatin bends and folds and loops and packs together closely until we can have the most condensed structure uh, of human DNA, which is the metaphasic chromosome. But we do not have this during interphase. During interphase in the cell cycle, DNA is not as condensed in the nucleus, and there are specific compartments in the nucleus preferentially occupied by specific chromosomes. And these we call chromosome territories. They are not static, they are dynamic, and they can be cell type specific as well and influence spatial organization and gene expression. Inside the chromosome territories, as we have illustrated here, we have smaller subdivisions called topologically associating domains for TADs, illustrated as these compartments here. TADs are self-contained sections of chromatin where sequences interact mostly and preferentially within themselves rather than with sequences from neighboring TADs. And TAD boundaries are defined by CTCF and cohesing proteins, and they form through the process of loop extrusion. where DNA is extruded in a loop through cohesing rings until the cohesing ring encounters convergently oriented CTCF sites. Um, and TAD boundaries can be disrupted, and that is associated with malformation and disease. For example, here you have a normal TAD boundary. If that with a CTCF site, and if that CTCF site is disturbed, you have a neo-TAD or a fused TAD as termed here. And this can lead to a phenomenon called enhancer hijacking, where an enhancer from one TAD influences a gene from another TAD, or what's supposed to be another TAD in wild type. And it's linked, for example, to polydactyly, brachydactyly, and a form of retinitis pigmentosa. And importantly, these conditions can happen even if the DNA sequence is unchanged, only due to spatial regulation. And we are looking to investigate the interleukin 8 TAD. So telling you a bit about interleukin 8 or IL-8, it is a chemotactic cytokine or a chemokine. Was the first chemokine discovered. So chemokines are cytokines are important immune system effector molecules. They are mainly secreted by macrophages in response to chemical or physical stress, pathogens, or other cytokines like IL-1 beta or TNF alpha. And it is the main driver of neutrophil recruitment and migration, and it promotes a variety of effector uh, conditions in neutrophils. For example, a prophagocytic state, the formation of neutrophil extracellular traps, upregulation of adhesion molecules, and buildup of reactive oxygen species. It also influences other cells. It might influence the migration of some other cells, for example, mast cells and myeloid-derived suppressor cells, and also the production of IL-8 itself by keratinocytes and macrophages themselves. And importantly, it also induces angiogenesis through the proliferation and survival of endothelial cells, and also the survival and migration of cancer cells. And that is important because in cancer and in inflammation, IL-8 is overexpressed. And here we have an illustration of that, where neutrophils degrade the extracellular matrix, and IL-8 promotes tumor cell survival and growth and migration through the epithelial mesenchymal transition. And that in turn prompts more IL-8 release. So there's the formation of the cycle. And we also have angiogenesis promoting more nutrition to the tumor. And that is why high IL-8 serum levels in cancer are often correlated with poor prognosis. It's also important in other diseases like cystic fibrosis, asthma, chronic obstructive pulmonary disease, and COVID-19, where it's involved in the cytokine storms. So it's very medically relevant. And talking a bit about the regulation of IL-8, its promoter has binding sites for NF-kappa B and AP1. So that links it to the broader immune response. And it's also regulated by a super enhancer region. And that is a cluster of enhancers that collaborate to regulate a specific genomic region. And we have that shown here. Recently, a new transcript was identified in MIA-PACA2 cells, which are an aggressive pancreatic carcinoma cell line. And MIA-link is a long non-coding RNA encoded inside the IL-8 super enhancer region, about here between umlilo and IL-8. And it is important because umlilo shares similar histone marks to MIA-link, and that might indicate that MIA-link may regulate IL-8, the high IL-8 levels in MIA-PACA2, because umlilo also regulate immune-related genes. So our investigation of the IL-8 TAD it was done with DCAS9 based tools. And what is DCAS9? It is a catalytically dead or deficient Cas9 enzyme. And Cas9 is an enzyme derived from the CRISPR, Cas9 is an enzyme in the CRISPR-Cas9 gene editing system. And that itself is derived from a prokaryotic defense system against viral infections. And what we have is Cas9 is a RNA guided nuclease. So we can associate with a piece of RNA, which guides it to a complementary DNA sequence. And there it has two nuclease domains where it's able to cause a double-stranded break in this target DNA. However, if we mutate experimentally these nuclease domains, we lose the nuclease function without losing the ability to guide it with an RNA. So, and that is exactly what dCas9 is. And because Cas9 N and C terminal regions are tolerable to fusions, we can use domains, protein domains, to fused with dCas9 and basically deliver it as a payload to specific genomic regions precisely. We have, for example, VP64, which is an activator domain derived from the herpes simplex virus, as well as CRAB, which is a repressor domain derived from zinc finger proteins. And this enables precise targeting of specific loci for gene modulation. So the goal of the study brings everything I've told you about together. We aim to do dCas9-based perturbation of the IL-8 TAD in MIA-PACA2 cells to understand IL-8 spatial regulation in MIA-PACA2, as well as elucidate TAD dynamics. The work I did mainly focused on isolating monoclonal cell lines of MIA-PACA2 expressing high levels of dCas9 CRAB. And we also aimed to begin our work in IL-8 TAD perturbation through the use of single guide RNAs to interfere with MIA-link and observe its effect on IL-8. So we started off with a polyclonal cell line of MIA-PACA2 dCas9 CRAB cells. We thawed these cells, we cultured them, we performed single cell sorting through FACS. Each cell was seeded into a different well in a 96 well plate. We let them grow as monoclonal cultures, and we selected the 10 most confluent cultures to grow separately in 12 well plates. We stimulated them with doxycycline because the dCas9 CRAB in the system is doxycycline inducible. That was done for 24 hours. And then we reserved part of these cells for RNA extraction. So we extracted RNA, we performed cDNA synthesis for qPCR, and then we performed qPCR itself, targeting dCas9 CRAB, as well as RPL37A, which is a ribosomal gene used as a housekeeping control. We did this in triplicates. However, we filtered out specific replicates that differed in CQ value by more than one from the median of that sample. And then we calculated relative expression of dCas9 CRAB relative to RPL37A through this expression. And we also calculated fold change compared to our unstimulated polyclonal controls through this formula, a delta-delta CQ approach. And these are the results we got. So we got a very clear winner, P2H2, and the names refer to the plate that they were derived from, so plate 1 or plate 2, and then the combination of letter and number that designates specific wells. We found statistical significance for the relative expression of P2H2 as well as P2H10 in comparison to the polyclonal control. We observed about a 15-fold increase in expression in P2H2 compared to the polyclonal control, and about 2.7-fold for P2H10. Then we went to Western blot to try to confirm this. So when we extracted RNA, we also reserved part of the cells for protein extraction, and we extracted protein through lysis with a RIPA buffer. Then we performed a BCA quantification to assess total protein concentration. Then we performed SDS page. We transferred it to a PVDF membrane. We did protein blocking. We incubated with primary and secondary antibodies targeting Cas9 and GAPDH, which is used as our loading control. And then we used, we did digital imaging through chemoluminescent detection. And what we got was interesting. We only got faint bands, and we got a faint band for P2H2 and a very faint band for P2H10, if you can even see it. Nothing else, no other clones had DCAS9 crab bands, even though they all had gap DH bands, so the experiment was probably okay. And also P2H2 had multiple bands, which was interesting. That was not exactly what we were expecting. So well, because we detected RNA for all clones and a lot of RNA for P2H2 specifically. So we decided to do everything again, culture all these clones again, and perform RNA and protein extraction and qPCR and Western blot again. And this is what we got. For qPCR, we got similar results. We had P2H2 as the #1 clone in expression as well, except this time the expression was 500-fold higher compared to the control. P2H10 came second again, except, well, at a very lower level. And all of the other ones were far lower comparable to the control. We this time got a beautiful band for P2H2, and we got a less pronounced but still very identifiable band for P2H10, and indeed no other clones produced bands. So talking a bit now about these results. Of course, we observed huge variability in expression of our gene of interest between clones, especially considering P2H2. And at first glance, it may seem curious how cells derived from the same virally transduced population, because these were lentivirally transduced cells, how they would have such variation. But what happens is each cell got the viral integration done in a different manner, right? So viral transduction is depicted here, and it is a complex process which can be influenced by many factors. And one of the most specific and one of the most important ones, sorry, is integration site in the genome, because the viral vector may integrate into active chromatin compartments, but it also may integrate in inactive compartments. And that can change expression in about 3 to 4 orders of magnitude, it has been reported. Copy number is also very important because the expression of the target gene tends to vary accordingly with copy number. And there can also happen gradual silencing of the viral vector by the cell through epigenetic mechanisms over time. Some other unexpected outcomes, one of them was our mismatch between RNA and protein levels, because we detected RNA for all cells, but we didn't see bands for most clones. And that happens because transcript levels cannot really indicate on their own protein levels because of processes like post-transcriptional and translational regulation, as well as protein degradation. So for example, processes like untranslated transcript accumulation and the protein half-life influence this. Here, for example, we have a graph from a study which shows the correlation analysis between mRNA abundances and protein abundances, and only 1 to 2/3 of the variation in protein could be explained by mRNA levels. And here we have human cells, a pie chart about human cells, telling us how much each of these factors could explain changes in protein. And mRNA concentration only explained about 27% of it. There could also have been the case that the proteins in the first Western blot were just too little to reach the detection thresholds for Western blot. And well, something else that bugged me at first was the fact that all clones survived and were resistant to antibiotic selection, because we performed antibiotic selection with them. We had G41A in the medium. And it was interesting because the resistance gene for G41A was in the same vector as the gene of interest. So how can it survive but not express the gene of interest? But what happens is antibiotic selection doesn't directly select for a gene of interest expression. It only selects for vector presence. And what can happen is that cells might only express the gene, the resistance marker, in enough quantities to only to stay alive, so minimal functional quantities, and not express the gene of interest in expressive levels. And there was also the fact that P2H2 had multiple bands in the first Western blot, and that might be due to assay artifacts, for example, insufficient protein blocking or excessive antibody concentration, or it could have been to protein degradation due to insufficient protease inhibition. Moving on, we performed immunofluorescence microscopy. We selected 6 clones, we seeded them on cover slips inside 24 well plates. We induced them with doxycycline for 24 hours, just like before. We performed fixation with formaldehyde. We incubated it with antibodies, 4009. We also performed Hooked nuclear counterstaining. Then we did the immunofluorescence imaging itself with the confocal microscope. And then we did image analysis. We did cell segmentation with CellPose, which is a generalist algorithm for cell segmentation. We measured the mean fluorescence for each cell in the images and normalized them to the background. And then we calculated the average of cell fluorescence for each image. And these are the results we got. And it was quite striking because P2H2 had the lowest signal, despite being the highest by far in qPCR and Western blot. And actually, clones that had little mRNA expression detected by qPCR had higher fluorescent signal. So that was inconsistent with Western blot and qPCR. These are our images. And here you can see that the DCAS9 crab signal is diffused across cells, which was interesting considering that it should mostly localize to the nucleus because of its nuclear localization signal. Due to all of this, we concluded that the signal for DCAS9 crab was likely non-specific. It could have been due to autofluorescence, but more likely it's due to non-specific antibody binding. We did use the same primary antibody here as in Western blot. However, success in one of those assays doesn't guarantee success in the other one because each assay has specific challenges. And one of such challenges in immunofluorescence is epitope masking, which might have happened due to formaldehyde fixation in our case. Well, this experiment likely needs to be repeated because it's very possible that something technical went wrong with it. We could also use the unstimulated polyclonal cells as negative control, which was not something that we did this time. We can also check for non-specific antibody binding, running the experiment only with the secondary antibody. And if the signal is really specific to DCAS9, it should fade if we only use the secondary antibody. We could also use small interfering RNAs targeting the DCAS9 crab mRNA for negative control, So employ RNA interference and then run Western blot and immunofluorescence in parallel, and then signal should disappear for both too. And another idea is to use mass spectrometry to confirm the presence of DCAS9 crab without reliance on antibodies. Well, moving on to our knockdown experiment, we wanted to try to perform gene knockdowns with single guide RNAs. We wanted to check the functionality of DCAS9 crab and observe in which single guide RNAs concentrations knockdown would be best achieved. And we also wanted to target MIA-link and observe if the single guide RNAs designed for it would be successful, and if so, what impact that would have on ILA. We chose P2H2 for this assay because it consistently showed high expression in qPCR and Western blot, despite immunofluorescence being discordant. So we got P2H2 cells, we cultured them, we transfected them with single guide RNAs targeting these genes, as well as scramble single guide RNAs. They were done separately, though, not together. So each single guide RNA went into a different culture. We stimulated them with doxycycline, of course, And then we incubated them for 48 hours this time. We performed RNA extraction and did qPCR. qPCR targeted these three genes, neolink, RELA, which is part of NF kappa beta, sorry, not beta, NF kappa B, as well as CCDC42B. And these two were positive controls. And we also targeted DCAS9 crab to check its levels, IO-Late, of course, to check for correlation with neolink, as well as RPL37A for normalization. And these are the results we got. Some of them were nice, some of them were not very nice. We did see stable DCAS9 levels. Oh, and by the way, the bars, the colorful bars represents the target of the single guide RNA used in each condition. So we saw a stable DCAS9 crab across all the conditions with different single guide RNase concentrations, 150 or 25 nanomolar, as well as with each of the targets of single guide RNase. And we saw knockdown for RELA at 50 nanomolar single guide RNA. However, we didn't observe knockdown for neolink, and knockdown did not scale with single guide RNA concentration. And as well as that, we didn't we weren't able to reliably quantify CCDC42B, so that's why it's not shown here. We performed correlation analysis as well between DCAS9 crab and the target genes, the other target genes, but we didn't find any statistical significant correlation. And one of our goals, which was to observe the correlation between neolink and IL-8, wasn't really possible because there was no neolink knockdown. We ran the correlation analysis either way. We didn't find anything significant, but the hypothesis cannot be discarded that neolink may influence IL-8 levels because we didn't see any knockdown. So we even checked the RPL37A CQ values to see if normalization could be a fault, but they were very stable across conditions. So it was likely not the case. We had this single guide RNA inefficiency, right, especially for neolink, and that can be due to structural features in single guide RNA. For example, depending on the structure, some guide RNAs can even preclude complex formation with Cas9 altogether. And target region also matters a lot because the closer you are, the closer the target is to the transcription start site of a gene, the more likely the knockdown will be successful. And we didn't know precisely the transcription start site for neolink. Even in the seminal paper that inaugurated CRISPR interference, which is the use of DCAS9 crab to repress genes, only 2 out of 7 single guide RNAs that they used in mammalian cells were effective to bring about some level of knockdown. There can also happen stress and immune responses in cells, which hinders the knockdown. And, immune responses due to the introduction of exogenous RNA, that is. And going forward, we can repeat the experiment to have more robust data and make sure that neolink knockdown really failed. We can try to design and use new single guide RNAs targeting neolink, especially if the transcription start site is elucidated. And another idea is to verify single guide RNA and DCAS9 crab complex formation to make sure that that is not the problem. That concludes our experiments. So talking about our main findings, we did establish different monoclonal Miapaka2 DCAS9 crab cell lines. We had pretty consistent qPCR and Western blot results, even if immunofluorescence was discordant from it. We consistently saw robust DCAS9 crab expression in P2H2 in Western blot and qPCR, so it was selected for functional assays. And we saw gene knockdown achieved for RELA at 50 nanomolar of single guide RNA. Concluding, we found that we have generated a robust monoclonal inducible DCAS9 crab system through the validation of B2H2 as our clone with strong and reliable DCAS9 Fab expression, we established a reference concentration for sgRNA use in the system at 15 millimolar. And this has the potential to be employed as an efficient platform for probing spatial regulation in genes. As some next steps, we have a few possibilities. We could repeat the immunofluorescence and single guide RNA knockdown experiments to have to validate the data, as well as have more replicates and explore the localization of DCAS9 crab again. We could design and test additional single guide RNAs for MiaLink. We could try to map the transcription start site for MiaLink for improved single guide RNA design and CRISPR interference. And then we also have to repeat this whole workflow for the MIAPACA2 DCAS9 VP64 cell line that we have, a polyclonal cell line, because this was only for the repressor domain, right? VP64 is the activator one. And then we can design and use new single guide RNAs to further perturb the IL-8 TAD in MIAPACA2 to gain further insight in IL-8 spatial regulation as well as TAD dynamics. That is it. Thank you very much for your attention.
Thank you, Lucas, for a very nice presentation. I'm gonna just quit the sharing of your screen, okay?
Okay.
If the evaluator wants you to rejoin it and share again, you are free to do that, okay?
Okay.
Okay, so as part of this process, I want to invite Professor Laszlo Groh for his comments and questioning, and thank you very much for your participation.
Yeah, hi. So wonderful presentation, wonderful work, Lucas.
Thank you.
Maybe I can ask, because this is a question and answers section, right?
Mm-hmm.
Yes.
I would like to ask a bit more details about the MIAPACA cells themselves. Of course, this was generated before you, but I'm just curious how they were generated and a little bit about whether you expect these to be DNA integrated sites or whether it's a vector introduced into the cell.
Yeah, so when I got there, they were already transduced, but I know that they were transduced with lentiviruses. And so I'd expect them to have integrated DNA because usually lentiviruses integrate into the genome, even though I think they can remain episomal, but the idea was to have them integrated.
Yeah, and this would also be the case for the antibiotic resistance.
Yes, yes.
Okay. So they'll be somewhat in the same locus being controlled by similar regulatory mechanisms.
Yeah, they were in the same vector, and then they were under different promoters, but in close proximity. So, yeah.
Okay. And you chose some pretty standard ways to validate this, of course, qPCRs, Western blots, immunofluorescence. Could you think of another way of validating that you have a good, stable knock-in in this case?
I mean, we could try to sequence them, but I don't think that would be the most efficient strategy. There is mass spectrometry as well to try to probe for the protein. And could we do fish with DNA? I'm not sure. It's been a while since I left Radboud, so molecular biology methods are starting to slip from my head. But these are some ideas that I can think of.
But I think sequencing is a good approach, especially, of course, in this case, it's a bit of a fishing expedition since you don't really have targeted sites, if I understand correctly.
Yes.
But it could tell you how it integrates it, and maybe if it messed up some of the genome along the way,
Yeah, yeah,
which of course is always a risk when you try to cut and manipulate the genome.
yeah. And also something that I didn't mention because of time is that apparently integration can also disrupt the vector. So it
Mm,
can integrate it in with an unintended form. So that could disrupt the expression of the gene of interest as well.
no. Yeah, which could explain some of the differences you see between some of the clones.
Yeah.
Yeah. All right, and maybe I can ask a bit about these pancreatic cancer and specifically IL-8 in that context.
Mhm.
Maybe you can refresh my memory on what IL-8 is doing to the pancreatic cancer.
I I don't know specifically about this type of pancreatic cancer where MIA-PACA2 was derived, but what IL-8 does is it recruits neutrophils. These neutrophils help degrade extracellular matrix as well as basement membrane. So there's tissue damage, makes it easier for cells to invade. So IL-8 also prompts this invasion, migration, survival of tumor cells, which causes tumor growth, as well as possibly the epithelial to mesenchymal transition and metastasis. It also can suppress part of the immune response through myeloid-derived suppressor cells and promote angiogenesis to nurture the tumor through the endothelial cell proliferation.
Okay. And the pancreatic cancer cells themselves produce IL-8. Do I understand this correctly?
Yeah, they produce a lot of IL-8. That's why partly why we chose them.
Okay. And what is the trigger for this IL-8 in these cancer cells, if there is one?
Mm, I wouldn't I wouldn't be sure. There I know that there is well the hypothesis that Neolink may upregulate it, though it's likely not the trigger itself. Um, but yeah, I'm not sure what would prompt IL-8 production, but yeah, it's common for tumor cells to produce IL-8, and then of course you have the uh, EMT, which generates a feedback loop and produce more IL-8. So yeah, that's as far as I would hypothesize.
So you think it's spontaneous self-producing IL-8 with no real inducer per se?
Yeah, that's as much as I can think because to be honest, I'm not really familiar with cancer biology that much, so wouldn't be sure about any specific mechanisms that drive IL-8 production in tumors. That's actually a good question. You got me thinking now.
Yeah, because of course we study IL-8 in the lab quite thoroughly, and we've come across a lot of different regulatory mechanisms. And I think in your report you also mentioned NF-kappa B, AP1. There's of course umlilo as another long non-coding RNA. There's a few
Yeah.
other long non-coding RNAs which are being annotated now, which also regulate IL-8. And
Hmm.
if you look at the transcription factor hub there, it's quite dense. So a lot of
Hmm.
things give rise to IL-8 at the end of the day. So as I was watching your presentation, I was kind of wondering maybe the fact you don't really see Mailer being affected by this guide is maybe not because of something technical, but maybe something biological. Um, and I'm wondering if the reason you're missing it is because it needs to be induced by a different transcription factor or a different signaling pathway. What are your thoughts on that?
Wait, I'm not sure if I understood everything completely. Could you repeat, please?
There are likely multiple discrete ways that IL-8 is being regulated in these cells
Uh-huh. Yeah.
based on, well, spontaneous production, as you think is happening, but also induced production by clear signaling pathways leading to a very uh, discrete set of transcription factors. potentially
Mhm.
mailers actually being regulated by one of these transcription factors. Do you think that's plausible?
What? Sorry, what is regulated by these transcription factors? You said
Oh, sorry, Mailink,
Neolink. Okay, okay,
referencing another
yeah.
gene.
So you're asking whether maybe Neolink is regulated by another transcription factor, and following from there, what is the question exactly?
Well, I was wondering if this logic follows for you, whether you think that this Neolink is actually being actively regulated rather than passively regulated.
Okay, okay, I understand it. Um, well, it could be the case, I think, because it's very Neolink is a very specific. It has a very specific histone marking signature for in Neolink, in Mia-PACA2 cells, hence the name. So I mean, it yeah, it does have more open chromatin in Mia-PACA2 in that region. Um, so I think, yeah, I think it could be possible that it's regulated by another transcription factor, but it is regularly produced by Mia-PACA2 cells. So I I'd expect even if it were regulated by something else, that it would be uh, regularly produced, and then we could knock it down
So you
annually.
think it'd still be sensitive to this DCAS9 approach?
I would, I think so, because DCAS9 crab modifies the crab domain modifies chromatin around it, right? So it prevents transcription factors, transcriptional machinery from reaching the gene as efficiently. So I would think even if there is regulation by another transcription factor, the use of crab would probably make it harder for this transcription factor to get in close proximity with the chromatin, and that would lead to uh, decreased transcription.
Okay. I think that logic follows. I think that's a good challenge of this pushback. I actually am very interested in what is figure 7 in your report. It's the immunofluorescence image where you show a couple of the different clones. I don't know if it would be
Yes,
possible to open up that slide.
I can open it's not in the present. Oh yeah, it is in the presentation as well.
Yeah, you can write
I
down the slide
can,
number.
yeah, it's okay. I can share the screen again, if that's okay.
Hmm.
Um, just one sec. Do you see my screen?
It's loading. Yes, perfect. Great. It's exactly the
Okay.
slide as well.
Yeah.
So maybe I can ask you what the regular morphology of these Mia-PACA cells is supposed to look like. Maybe just
Yeah.
pointed like a regular one of the panels that looks the most similar to a wild type.
hmm It was interesting as we were growing them because, well, in pictures from biotech companies, for example, that sell them, they're usually more like this, more elongated. They have these protruding edges. But
Mm.
as we were growing them, we saw both this type of morphology as well as this more rounded one. And in fact, P2H2 is this one, and it had more rounded morphology. So I'm not sure how morphology would affect that, or how what it would indicate, because
Mm.
that's pretty that's really out of my field, I think. But it it does look somewhat like this, but these we did find them to look a bit strange as well during microscopy, also because they were clustering a lot, and these were some of the clones that didn't have high DCAS9 crab transcription. It might be a coincidence, but yeah.
Yeah, but you agree with me that the morphology for your preferred one, P2H2, so panel C, looks a bit funky compared to what's on ATCC.
Yeah,
Yeah.
yeah, it does.
Okay.
It's interesting.
Yeah, and the signal intensity was relatively low, but in this panel, it's very green, and green, of course, is your protein of interest. So this is
Hmm.
the DCAS9 crab.
Yeah.
But if I look for the DAPI or the Hoogst, the blue,
Hmm.
it's almost absent in these cells, or very hard to pull out. Um, can
Yeah.
you maybe give some background on why that appears to be the case?
Oh, I assume that is just due to the overlay with DCAS9 signal. Um, but I'm not, well, it was diffuse, right, through the cell. And I'm not sure that it was actually DCAS9. So I think it could have it could have been just because the DCAS9 signal was very uh, pervasive, and then it interfered with visualizing the DAPI signal. But also, it is quite green, but we did perform like different. We adjust the contrast and brightness while analyzing the images, and also while generating these images for better visualization. So I'm not sure
Okay.
how much it reflects fluorescence in like an absolute level.
And indeed, that was a technical question I was going to ask you. Is every panel here using the same intensity settings for the different colors?
Yeah, so no,
Okay.
they're not. They were just we just selected automatic adjustment in ImageJ just to make these just to make them visible, mostly for illustration of the distribution and the morphology.
Yeah. All right. All right. So I shouldn't take too many strong conclusions from what I'm seeing with the lack of nuclear staining here, and
Yeah,
focus more on the bar charts.
yeah, I think so. There's also, I mean, I couldn't include it on the thesis, but there are the sheets with the values from each cell. So that would be probably more reliable if you
Hmm.
really want to look at each cell individually.
Yeah. And just going back to this morphology effect,
Hmm.
what do you think could give rise to this morphology, especially with the intervention that you did?
Um, well, we added G418 to the medium, then we added doxycycline to the medium. I'm not sure how those interfere with morphology, but I would think they since they're antibiotics, maybe they actually I'm not 100% positive that doxycycline is an antibiotic, but I think so.
It is.
Uh, it is okay. Yeah, I'm guessing that probably has effect on morphology. I,
Okay.
I
And
would
why not
think so.
for the cells in panel B and D if it is the antibiotics?
True, good question. May maybe it has to do with resistance gene expression. Maybe they express different levels of the resistance marker, and that somehow links to the morphology. But I wouldn't be sure. I was puzzled as well by how the morphology here, these two morphologies that we observed, link to anything else from the data.
Yeah. And going back to what we were talking earlier about different approaches to kind of validate your CRISPR approach,
Hmm.
do you think that sequencing could help give clues as to why you might have changed your morphology?
Mm, I, well, I think so. If if it does have something to do with resistance gene, uh, then we could see how it's distributed and how it's integrated in the genome, how many copies there are, if it's in a regional alternate closed chromatin, and that might elucidate a bit about, well, that might uh, help us infer something about the expression, and maybe that can link back to morphology.
Yeah, but the integration is also random, so there's also a chance that
Yeah.
it's something critical, right?
Hmm. Yeah, that's true. Yeah, so there could be the chance that it just integrated and disrupted a gene somewhere, even though genes are a very small part of
Hmm.
of the DNA. Like, I guess, yeah, there is a chance that it disrupted uh, some gene, although I wouldn't think that there would be a huge chance that it disrupted the same mechanism in all of these cells that are more circular, um, because it would be too much of a coincidence.
Yeah.
But yeah, but it is interesting that we observed the circular morphology for about half of the cells, and then the more elongated one for the other half, and that usually the ones with the elongated one showed less gene of interest from that production.
Yeah. Actually, speaking about that, talking about different clones and how they might be affected differently by the treatment, you
Hmm.
have two which gave some signal for the Western blot at the very least. And I'm
Yeah.
just curious why you didn't choose to use that for the same downstream assays to do the guide transfections in there and see how that would affect the knockdown of Mhlanga.
You mean why I didn't use B2H10 as well?
Yeah, yeah, that one exactly.
Mm. Yeah. Okay. Well, to be honest, I think that was mostly due to practical factors and time constraint, because it was close to my time to go back to Brazil when we were doing that part, and we just we just assumed, well, since P2H2 shows this huge difference in comparison to the other ones, it is more likely that there's going to be more functioning decazing crab. So, and that's what we had time to work with, really. But I think that would be a sensible approach to try to replicate it with other clones, especially B2H10.
Yeah, very fair. And for the immunofluorescence, do you just have a single replicate, or did you do this multiple times?
We, yeah, I didn't mention that, but we reported 3 images from each clone. These are just the most, the ones that were, well, the prettiest to illustrate it. But
Mm.
but we did take 3 images and then use those 3 images in the analysis.
Yeah. So when you say 3 images, are we talking about one well where you take 3 different fields of view, or were you talking about 3 different wells that you did this treatment?
Oh, yeah, okay. No, it was just one well.
Okay. Okay.
Yeah.
And you didn't take along a wild type control, just of healthy Mia-Paca cells.
No, yeah,
Okay.
that's the thing that I mentioned, and that thinking about it afterwards, I was like, hey, we should have used the polyclonal control for that as well.
Yeah. Because that would be really enlightening in view of this morphology, right? If these treatments
Yeah,
themselves would induce this. Yeah.
yeah, exactly.
Of course, you would expect no staining as well for the Cas9, which would tell you quite a lot about, um, you know, whether the antibody specific.
Yeah, yeah. I think that was more of an oversight, really. And then we just took the clones and did it with the clones, but no negative control.
I'm curious how much time I have left.
Oh, you have 2 minutes, but if you need a little bit more, it's okay.
Okay, I'll then just ask one very quick question. So you argue that you might lose some of the the expression of these Cas9 proteins in these clones because you cultured them for a long time.
Mhm.
What would kind of drive that process?
Well, from what I've been reading, there can be simply a stochastic silencing effects from in the cells, and then histone histone methylation around that site could lead to silencing of the vector. These through epigenetic mechanisms. I don't know if it would have something to do with the fact that it's a vector. I'm not sure if there's a way for the cell to detect that, to tell that apart from regular DNA. Maybe there is, and it's slipping my mind. And then it has something to do with that. But yeah, as far as they can tell, just those stochastic effects from epigenetics.
Yeah. And how would you go about like trying to test that, trying to validate that? Because of course it could interfere with your attempt to knock these genes down.
Mhm. Um, I mean, we could try. We could try to do. It's not. I don't know if it's chip sync. No, it's not chip sync, but something in my mind that's in the lines of um, trying to get the histones that are close to the vector, and then checking for their histone mark signatures, and then maybe that can tell us something about whether these loci were genetically silenced or not.
I think that would be a valid approach. Yeah.
Mm.
Okay. I think I've asked most of my questions, so I'll give it back to you, Lucia.
Thank you so much, Laszlo.
Thank you very much, Professor Laszlo, for your comments and interesting
Doctor,
questions as
please.
well. Okay, Dr. Laszlo. So now we are going to pass to the phase in which I will open for the supervisor to do some comments and wrap up and something like that. Dr. Musa, yeah, thank you very much. And you can speak if you want.
Yeah, so I guess I should kind of give some overview and some comments on what I saw from Lucas' time working with us in the lab. And I guess I just want to start off with today. I think, you know, everybody should know that the work that Lucas has done here in our lab for his undergraduate thesis is very similar to work we ask master students to do here in the Netherlands. So already this kind of a project, he's about 50% of a master's student thesis, just so everybody is clear who's listening here. So I try to calibrate my remarks and my comments and my evaluation based on that benchmark. So clearly, from the point of view of the expectations of the system we have here in the Netherlands, Lucas is far exceeding that. We do not expect bachelor students to do these kind of very complex experiments. So for this, I really want to say, well done, Lucas. Congratulations. Biology is an experimental science, and experiments don't always work. Laszlo put you through your paces and really, expose some of the many reasons why you didn't get the desired results you'd want to, but it's biology. We're dealing with an experimental system. But you should be commended really for all your efforts over this time, and so that's a good departure point for me, because the way I would like to contextualize Lucas is that I've worked a lot with colleagues and students from Brazil, and I've been to Brazil many times to teach different classes, never to undergrads, mostly to PhDs and postdocs. I've found the PhDs and postdoc students always to be outstanding, and the trainees are very well trained. And Lucas is just an example of that. And so I'm always eager to have students, postdocs from Brazil in the lab. And I've had a lot of interactions with students from, especially from Sao Paulo and from this city outside Sao Paulo. I've mentioned it to you before, where there's the accelerator, like
São
the
Carlos. São
like
Carlos?
no, there's like a
Campinas.
Campinas, where there's like a synchrotron. I've taught a class there. I've had people from there, so always great. Lucas came to the lab in a very interesting way. He was I mainly teach master students, and but I do teach 2 sessions of an undergraduate course on functional genomics, and Lucas was a student in that course, and he was very interactive, and he was really great at answering questions. And he came to talk to me after class one day, and I said to him, Lucas, you know what? You seem like a smart guy. Why don't you do an internship in our lab and do some work at the bench? And Lucas jumped at the opportunity, and that's how he came to us. And I think today he's, come to the end of that journey by really presenting what I think is a very well structured and, in many parts, conceptually strong thesis in a very complex area of advanced genome regulation and CRISPR-based gene expression modulation. There's some aspects of his experimental approach which are very difficult and could be improved, obviously. But that happens to people who are like me, who are supposed to know how to do these things, and we still have those issues. So I wouldn't fault him on that. But I think he definitely demonstrates a very advanced level of scientific reasoning and maturity in this sense. He also demonstrates the independence and ability to do data interpretation and problem resolution that's also far beyond my expectations for master students and his peers here in the Netherlands. Now in Brazil, where there's a lot of smart people, I don't know, maybe he's like in Brazil, he needs to meet an even higher standard. The other thing too is that I think, in terms of the way the manuscript is written and the figures are done, it's well organized, it's clearly written, it really demonstrates, thoughtful and honest interpretation of, both the technically challenging results and the ones that are actually working. And if it was up to me to ask questions, I wouldn't have asked the same questions that Professor Laszlo asked, but I would have asked different questions to kind of help you to go more into problem resolution. So in terms of your oral presentation today, I really thought you did a very, very good job in presentation. I think you did a really good job in fielding questions from Professor Laszlo, and I think that you definitely are deserving of your undergraduate degree and your thesis. So from my point of view, um, Lucas, you've fulfilled, a great deal of the criteria for what is needed for your thesis.
Thank you so much. That is very nice to hear, Musa. I'm really glad.
Oh, can I mention one last thing? One last thing, which
Sure.
has nothing to do with the thesis or the work. I just want to say that Lucas has been a wonderful lab citizen. He's been such a great part of our lab community, which is very international, and we were really happy to have him as part of our lab family and for him to participate in so many of our work and play events. And we're very, very sad that he's going back to Brazil. Please send us somebody else like Lucas to come to us. Please, please, please. Por favor, por favor. Obrigado, Lucas.
Dr. Musa, yes, we want to send more people, and we have some Lucas friends and colleagues following us on YouTube, and then they know they are invited, and it's really going to be a very good experience from them. And but they come back, okay? They need to come back to to stay here with us as well. Okay. Thank you so much, Professor, um, for the opportunity that you gave to Lucas, because it was not even supposed to be an undergraduate final project, but Lucas took the chance and embraced it, and just it made most of that. Okay, so thank you very much. I will now open the talk to Dr. Élida if you want to do some statements as well.
Good morning to everyone. Well, I also want to say all the best to praise Lucas because he was really a gift. He is a very hard work person. And actually, I'm not involved in this work. I didn't take part on that. I'm here just as an ex-supervisor for Lucas when he was here in Brazil before he go to Netherlands. But it's a great pleasure to see how much he had progressed, and he's a person that take all the opportunities that comes to him, and you can see how he took this opportunity to grow even more. And as Dr. Musa said, he's a very easygoing person. He had the best of relationship with everybody in the lab. And while he was in my lab, he worked really hard. He was very hard worker. And so I'm very pleased to see. And I'm sure he's going back and he's going to shine. And I would like also to take the opportunity to congratulate his parents that embraced all his dreams and make his dreams to come true. And I think that's is a very important to have parents that support you like your parents did. And I'm really proud. I didn't take part on this job. I just thank Dr. Musa to have him and to have done such a good work. You can see, I think his presentation, the writing, was everything perfect. I'm really proud of Lucas, and I wish all the best for you. And that come back sometimes to visit us.
I will. Thank you so much, Élida.
Thank
Thank
you.
you very much, Dr. Élida. So, Lucas, if you have any few words to share so we can wrap up this session.
Yeah, of course. I'm I'm very happy with the work I did. I'm very happy with the opportunity I had. I want to thank Musa very much for taking me in, for giving me the opportunity, for uh, yeah, helped me settle in the lab and for enabling me to use this as a bachelor's thesis. Because yeah, I didn't at first I didn't even think that my work there at the lab could be used as my thesis. I thought I would have to come back to Brazil and then do some other work and spend a year more here at least. And it was so great to have the experience there and also couple that with uh, my thesis. Um, so that was really all of it was a life-changing experience. The lab, the Netherlands as a whole, was a life-changing experience. And I want to thank you, Musa, for being a part of that. I want to thank Élida as well for having been my supervisor previously, for giving me the opportunity to start off in science and bench, for your input in this thesis as well. Um, and also thank you too for all the kind words. That it really means a lot. It really makes me very happy to make people who I take inspiration from proud. And also thank you, Laszlo, so much for being down to do this, for asking such insightful questions, uh, and for yeah, being very encouraging too. And also you, Lucia, because without you, I wouldn't have been able to have this online presentation, to use this as my as my bachelor's thesis. So yeah, you know how we've been coordinating ever since last semester to try to make this happen. So that was very meaningful as well. And I want to take the opportunity to thank my parents as well. As Élida said, they are very important to me in my life. They're very supportive. They enabled me to go to the Netherlands, have this experience as well, and I hope I'm making them proud. And finally, I want to thank, well, oh, actually, one more person. I want to thank Moomin. I want to thank Moomin for having been an amazing supervisor. He's supposed to be watching. I hope he's still watching. It was such a pleasure to work with him. He's such a joy and so helpful and so diligent. He was really an inspiration too. And finally, thank everyone who's watching, all my friends, my family. Um, I'm really happy that you could take some time and give me a bit of your attention to maybe hear about something that you know almost nothing about, just to be here for me. That makes me very, very happy as well. And if you're a friend and you've been watching, please send me a message to know what you thought about it. And yeah, just thank you everyone for the opportunity.
Thank you, Lucas, and I just want to congratulate you on this well done job indeed. Okay, and I think you took most of this experience, and yes, this is really important because you were so far away from your family. I was wondering how many months did you spend in the Netherlands? Did you do this in 1 year?
Yeah, so that's always funny when people ask me. I was supposed to have stayed 1 semester, and then I
Okay.
liked it way too much, and I extended it to 1 year. And then I was like, yeah, I don't want to go back yet, so I extended it once more. So in the end, it was 1.5 year, and the internship itself, it was just a little over 1 year.
Okay, okay. So congratulate on that, and
Thank
you
you.
know, way to go, and the sky is the limit. Okay, just I wish you all the best in the upcoming opportunity that you might have. Okay,
Thank you so
so
much.
I just want to say thank you to everybody who's here, just taking this a little bit more than an hour to evaluate Lucas and share comments, very nice comments. Thank you so much. And Dr. Musa and Dr. Laszlo, you are always welcome to be supervisors as well. of our students. It will be an honor to share the screen with you again. I wish we had money to bring you here to get our coffee and cheese bread
I
in
offered to
person.
come. I
You
offered to
know,
come.
sure, but if you are invited, whenever
No,
you
no, no,
have the
no, no, no.
chance.
I was gonna pay myself. Don't worry.
Yeah, okay, cool. So, but whenever you decide to come here to Brazil, just take a look and come here to the Federal University of Minas Gerais. It will be a pleasure to have you here, okay? So, thank you very much, everybody who's also watching us on the YouTube. So, I will just end this broadcast, and you have a
Congratulations,
nice
Lucas.
day.
Well done. Congratulations, and I'm so glad that working with Moomin and Laszlo and other people in the lab, you've done so well, and you have amazing supervision from Élida and Luzia. So congratulations again, and
Thank you so
the very
much.
best. All the very best.
Thank you,
Okay,
Musa.
so have a nice day and nice almost evening, I guess, in the Netherlands.
Mhm.
Okay, bye bye.
Bye.